Hello, everyone. Renovo Neural would like to welcome you to exploring disease-relevant readouts in Parkinson's disease models. I'm Allison Troughton from Renovo Neural, and I will be moderating today's webinar. I would like to introduce our presenter today, Dr. Zuza Kurowska. Dr. Kurowska is a lead on the development of preclinical models for testing anti-Parkinsonian therapies at Renovo Neural. She has extensive experience in PD research, including transplantation therapies and complex genetics. Please feel free to type your questions into the chat at any time during the presentation, and we will address them in our Q&A session following the presentation. Take it away, Zuza.
Thanks, Allison. Hello, everyone. First, just want to let you know that this presentation will be illustrated by a combination of our internal work and published data from us and others. Also, what you will hear here is an example of how a Polish accent sounds. In the next half an hour, I plan to take you on a journey into the world of preclinical drug testing in Parkinson's rodent models. First, to put everyone in the picture, we will touch upon Parkinson's disease per se. I will briefly discuss an unmet need when it comes to PD treatments and how PD rodent models can be employed in symptomatic and disease-modifying therapy development. Finally, we will focus on a palette of 6 preclinical endpoints helpful making strategic decisions whether to move on with or drop a potential anti-Parkinsonian drug. Our goal is to understand the translational value of those 6 types of analysis in developing novel therapies in PD. So let me start by saying a few words about Parkinson's disease. It is estimated that in the US, 1 in 100 people over age of 60 suffers from PD, and about 60,000 new cases a year will be diagnosed. PD is recognized mainly on its motor symptoms such as tremor, rigidity, or balance problems. Lesser known non-motor symptoms include depression, fatigue, gastrointestinal problems, or dementia. The majority of cases, about 90% are sporadic, caused most likely by complex interactions of gene and environmental factors. The rest, about 10%, are familiar forms of PD, and they are attributed to mutations in genes such as alpha-synuclein, LRRK2, or GBA. Knowing that the best Parkinsonian drug, levodopa, that has its own serious side effects, was discovered 50 years ago, one can surely appreciate the burning need for the new therapies. It is true that there are several other treatments available, such as dopamine precursors and antagonists or MAO-B inhibitors. These are symptomatic treatments targeting only motor symptoms, and they work just for limited time, which leaves unmet needs for patients and physicians. The acute toxin models, such as 6-OHDA, MPTP, or rotenone, have been helpful in development of those motor symptomatic treatments. At this time point, the key challenge for drug development is that lacking efficient biomarkers, the PD diagnosis takes place much too late, and it relies most solely on clinical motor symptoms. With the novel biomarkers and precision medicine approaches, hopefully soon we will be able to recognize the Parkinsonian phenotype much earlier and with much higher precision. For that new era in PD drug development, most useful will be genetically modified rodent models with clinically relevant mutations. In contrast to toxin-induced, those models usually display a long and gradual prodromal phase. The analysis here will be focused on specific molecular pathways involving molecules such as alpha-synuclein, LRRK2, GBA, Parkin, and others yet to be discovered. No matter which PD models are favorable, one has to pick carefully from a smorgasbord of various in-life biochemical or morphological readouts available. In this part, we will focus on the translational value of the 6 readouts most commonly used with PD rodent models. Let's start with the longitudinal methods. Behavioral tests are very helpful tool in preclinical drug testing in PD because of their predictive value. Without having to sacrifice an animal, one can have an idea about pathology progression. While there is a variety of motor and non-motor tests to choose from, historically, especially useful behavioral tests were ones that measured dopamine-induced motor deficits. Here, for example, the rotations in apomorphine-induced rotation tests were measured after unilateral Nigel 6-OHDA lesion. The amount of net contralateral turns was correlated with the percentage of dopaminergic striatal denervation present after the lesion. This and similar tests can be a useful tool to exclude from the experiment those mice that do not have sufficient lesion before the drug or vehicle groups are formed. Focusing on the prodromal phase of Parkinson's disease, impaired olfaction can be early sign of PD and can be observed 2 to 5 years before the motor symptom onset. Alpha-synuclein accumulation or aggregation and altered neurogenesis may play a role in this phenomenon. Here in this study, testing effects of 8-week Resadgelin treatment in F28 alpha-synuclein in wild-type mice, a battery of olfaction behavioral tests was employed. The results show that the olfaction deficits, such as inability to appreciate the social and non-social odor by transgenic mice, were ameliorated by Resadgelin. In general, behavioral tests have been and still are the go-to readouts when evaluating anti-Parkinsonian treatments. A newer in-life technique that in the future can have great implications in drug development is MRI. Magnetic resonance imaging is a non-invasive in-life technique with potentially increasing translational value in preclinical testing in PD. It gives ability to evaluate and compare the anatomy or physiological processes in the healthy and diseased brain. Over the last 10 years, the MRI potential for evaluation of progression of Parkinson's disease in the clinical setting is increasing, and high-power MRI scanners are employed for the animal models as well. For example, it was shown that free water measurement different brains of patients with an early PD from the healthy controls, and similar MRI scanning modality was employed in the Parkinsonian mouse model of early onset generalized torsion dystonia. In the striatum and in the cerebellum, significant increase in free water was observed in transgenic mice compared to the wild type, here indicated in orange. And summing this part up, the main advantage of those two in-life readouts is their relatively non-invasive longitudinal character. We made the most of in-life techniques. Now it's time for tissue collection. But how we will evaluate the animals on biochemical level? Two classical methods for understanding the neurodegenerative diseases are HPLC and Western blot. Dysregulation of neurotransmitters, especially dopamine and dopamine metabolites, appears early on in the PD patient striata. This is a sign of malfunction and degeneration of synapses, axons, and the dopaminergic neurons of mesostriatal pathway. Same phenomenon is induced in most of classical toxin-based and some genetic PD models. So how do we measure that? We will employ high-performance liquid chromatography, HPLC. HPLC gives a unique possibility to distinguish and measure amounts of molecules that have almost the same chemical structure. As an illustration to this, here's an engrailed 1 heterozygous mice, and both dopamine and its metabolite DOPAC were decreased from 4 to 24 weeks of age. And at 4 weeks, long before the dopaminergic neuron's death appears, the metabolism of dopamine was increased suggesting some early sort of prodromal synaptic changes in this model. Similarly, in the other study, the intoxication with MPTP evoked the loss of striatal dopamine and DOPAC as well, and the treatment with MSDC drugs increased the amount of those neurotransmitters significantly. The key part of Parkinson's is deregulation of biochemical equilibrium of the brain and maybe even other tissues. Aside of neurotransmitters, abnormally down or upregulated become also proteins such as enzymes needed for dopamine production, synaptic proteins, including alpha-synuclein, and mitochondrial and autophagy markers that can all be detected and quantified with Western blot. One of those downregulated proteins is tyrosine hydroxylase, a rate-limiting enzyme in production of dopamine. On the left, you can see here a Western blot for TH and a housekeeping gene, beta-actin. There are 3 wild-type and 3 engrailed 1 mice at each time point. When this is quantified, we see significant changes at 4 weeks, down to 50% loss at 24 weeks. And similar observations were made in dopaminergic neural neurons of people with PD. Many enzymes are regulated not only through their expression, but also via their post-translational modifications. LRRK2 is a kinase which bears mutations that cause or contribute to PD. As many kinases, it is regulated by phosphorylation. Kinase inhibitors are new potential treatments in PD models, and antibodies against special phosphorylation sites can help assess bioactivity of those enzymes. Here, the expression level and phosphorylation of LRRK2 kinase was assessed in transgenic mice. Both doses of the treatment, 3 and 30 mg per kg, significantly reduced the ratio of phospho and to total LRRK protein. And at the same time, the total LRRK2 protein was not reduced significantly in either tissue compared with the vehicle control. And this altogether points at decreased bioactivity of LRRK2. While Western blot can give you quantification, immunohistochemistry, IHC, can provide more contextual information about your protein. And let me use a couple of examples here. Let's start with the classical in PD field TH staining. Striatum, that is a relatively large structure, is innervated by axons of TH-positive dopaminergic neurons that have their cell bodies in the substantia nigra. In PD, those cells downregulate TH and degenerate, which also is evoked in several Parkinsonian rodent models. In a striatum, TH axons can be very easily quantified from multiple slides using automated image analysis. Here I'm an example of 6-HDA unilateral model. On the other hand, the state-of-the-art method when you would want to use for TH cell body quantification, for example, when using the MPTP model, is serology. This method includes systematic sampling in the defined region of interest in the fraction of sections. It is a method with high accuracy and virtually no bias when applied correctly, and it can be used to estimate the total number or length of any cell population in a region of your choice. For example, the total length of blood vessels or neurites in a graft, or total number of mitochondria in midbrain. In this slide, we see a sham and lesion rats with IBA1 staining that visualizes microglial cells in a small pilot study. The total number of IBA1 positive microglia in midbrain is quantified in IPSI and contralateral side of the lesion. And we can see that after the toxic insult, there are somewhat more microglial cells present in midbrain on the injected side versus the contralateral site, suggesting increase in microglia proliferation. Zooming in even further, when using IHC, one can ask questions about the alpha-synuclein conformation and its cellular localization. Alpha-synuclein misfolding into amyloid sheets and accumulation in form of Lewy bodies are key hallmarks of PD. There are a few reliable alpha-synuclein rodent models that recapitulate those phenomena. Here, for example, stained in red, we see alpha-synuclein. It accumulates in cytoplasm and neurites of neurons of mouse injected with alpha-synuclein preformed fibrils. Thioflavin counterstain in green is the marker of beta sheets seen in Lewy bodies in PD patients. And it colocalizes with the alpha-synuclein staining here, indicating that alpha-synuclein forms amyloid fibrils. We've gone through morphological quantitative readouts using IHC in different ways. But what happens if you want to increase resolution even more when looking at morphology of cells? The next level is electron microscopy. And here I'm sure many will agree on this with me, that looking at the electron micrographs when you haven't seen tons of them can be confusing. For me, the question always remains, like, what happens beyond this snapshot here? Does this mitochondria look like that, like 1 micron away too? Well, one invaluable technique here is 3D EM. Structural changes in organelles such as mitochondria, phagocytes, as well as synapses are seen in PD brains and also in animal models of PD. Three-dimensional electron microscopy, called also the serial block-based scanning and electron microscopy, is a great tool to use when going into the cellular organelle resolution. This technique involves an automated process of sectioning, imaging blocks of tissue, and data acquisition. No other technique can provide such an accurate information on number, volume, size, and distribution of organelles, for example, mitochondria, in a defined brain region. Autophagy problems and axonal trafficking changes differ PD from non-PD brains postmortem. In many animal models, those malfunctions are also observed. For example, different forms of axonal viscosities in striatum of engrailed 1 heterozygous mice at 2 to 8 weeks of age were identified using 3D EM. The colors in the lower row indicate the progression of the pathology. You see yellow, that is early stage with only few dystrophic organelles visible, blue, intermediate stage, and navy blue, late stage with multiple electron-dense bodies. Going into subcellular level, mitochondrial changes are fundamental to various disease conditions. Neurons in particular are sensitive to mitochondrial changes as they transmit and receive electrical signals to and from distant locations. 3D EM gives a unique opportunity to quantify those changes. Here, for example, in this neurodegenerative disease model, we see the swelling of mitochondria in red. Those abnormalities were observed in a 3-week-old animals and was almost 6 months before the neurodegeneration was detected. These mitochondrial changes that we can readily quantify suggest probably lowering of metabolic capacity and degeneration. Similar abnormalities are most likely present in many years before the Parkinsonism develops in humans, and also early on in some transgenic rodent models. Today, I've taken you through different methods with high translational impact in drug development in Parkinsonism. We talked about the longitudinal changes. They can be monitored by behavioral tests and with MRI throughout the course of the disease. Then we touched upon measuring the biochemical imbalance of molecules and their pathways using HPLC and Western blot. And finally, we discussed the evaluation of some cellular and subcellular parameters in Parkinsonian brains with methods such as immunohistochemistry and 3D EM. In conclusion, this table should help summarize our presentation today. Here at Renova, our approach is to build high-quality, reliable studies with the most appropriate disease readouts to facilitate rapid preclinical decisions. Two potential scenarios could be considered here. If you already have a PD model or study at hand, we could work with you to examine your precise mechanism of action based on what specific phenotypes you are targeting. Alternatively, if you believe your drug has a potential PD indication, we may build a study with broader scope to encompass several different disease-relevant readouts. These approaches will help to reach a strategic decision at optimal cost of time and money. Whichever model is chosen for therapy testing, we need to learn from it rapidly. In other words, it is of everyone's benefit, especially the researchers involved in discovery and translation, to evaluate each new treatment opportunity comprehensively and in a timely fashion before advancing into clinic. And how to do that? As a preclinical research service provider, our role is to help advance the development of potential new therapies and help to identify molecules that have potential for translational success. At Renova, we can partner with you to come up with the right strategy and tools to achieve your discovery and translational goals for PD. Thank you, everyone, for joining this webinar. I would like to also thank all my colleagues at Renova who helped with shaping it. Now there will be time to ask questions, but just wanted to let you know that in case you have some questions that come up later to you or some afterthoughts, here's the address you can contact me at. This is vkurowska@renovoneural.com. Thank you.